Hello, my name is Darren Mueller, Extension Plant Pathologist at Iowa State University, and today's talk is going to be on soybean diseases. And so we're really going to be covering two of the major soybean diseases that we saw in Iowa in 2023, white mold and sudden death syndrome. And for white mold, it's going to be a little bit different because it's not around management. It's going to be the question is like why we saw so much of it in sort of a unique year. We saw it in fields that we typically haven't seen it in. And so we're going to sort of dive in a little bit into some of these questions that came up throughout the season. And then there's going to be an SDS management update after that. All right. So to better understand sort of the answers in the Q&A, we're going to quickly go through the disease cycle for white mold. And so this is a fungal disease. It's caused by Sclerotinia sclerotiorum. And it is, let's start with the letter A on the slide. If you look at the very bottom, and so these are little black structures, are called sclerotia, and this is how the fungus survives. And if you look at the photo up on the top left, you can see the little black structure, and then there's little mushrooms coming out of it. But so the sclerotia, that's the survival structure. It can germinate and produce stipes. That's what you see in the letter B. Or if you look really closely at the detail, there's some white growth on one of those sclerotia in the letter A, and that's actually just mycelia. And so that's the fungus. That's where it gets the name white mold. And so it can germinate mycelial growth, or it can germinate carpogenically, and that's the letter B. Those are the stipes. And so let's go with carpogenic germination. If you go to the letter B, you can see the stipes, and then eventually those stipes will turn into apothecia, which is what you see at the letter C, or you see those in the photo on the top left. And these apothecia they're just small little mushroom-like structures that produce lots and lots of ascospores, which are the spores that will eventually infect the soybean plant. And so, rewind, if you go back to the letter A and look at the mycelial growth there, this type of growth will stay in the soil, but it has the ability to then either infect roots of certain plants or possibly grow saprophytically, which is grow on dead tissue and then produce more sclerotia. So it's a way to sort of survive for over a longer period of time in the soil for the fungus to survive multiple generations. And so it is fairly complicated, and there's a lot of detail that sort of goes from the letter A to letter B, and then letter B up to an apothecia, letter C, and we're going to get into that when we answer some of the questions. But eventually, the ascospores that you see in that top part of the inset in letter C, those will land on soybean flowers. The soybean flowers need to be senescing or dying, and those will eventually get colonized. And that's the infection point and sort of the how the fungus gets into that plant. And then eventually you will get the white growth, the white mold, where it gets its name. You see in letter E, and eventually more sclerotia are formed at the end of the year, and those sclerotia are either inside the stem or outside the stem, and those drop back into the soil, and the cycle happens the next time soybeans are grown. So that's in general the life cycle, but there's some details in there that we're going to tease out as we answer some of these questions. And so the first question was, why did I see so much white mold in new fields? And so I think the question I got several times this year was, did I plant a bad batch of seed that maybe had the fungus? This fungus can be seed-borne, and so and also the sclerotia, sometimes if it's a bad enough infection, the sclerotia are actually about the size of seed, and sometimes those can be in a seed bag. And so you could just be planting the sclerotia, or you could be planting infected seed, and maybe that's why we saw so much white mold in new fields. And also, there's a lot of more equipment that's being shared or sort of going from field to field. Maybe we have a lot of the sclerotia or the infected material that is being moved from field to field on equipment. And so the answer, I would say, for this one is that if you planted infected seed in 2023, this is probably not the source of white mold in 2023. And the reason I say that is that yes, you can go from an infected seed, and then that infected seed will actually start to germinate, but then it'll quickly be colonized with mycelia, not the apothecia, with mycelia, and then eventually a couple of sclerotia will form where that infected seed was at. So it sort of replace, you get infected seed and is quickly replaced with a couple of sclerotia. But sclerotia typically need to have some kind of cooling period before it will produce an apothecia. So there's only one report right now in literature that shows that you can go from sclerotia to apothecia in the same year, and those apothecia really were not produced until really late in the season, like all the way until September. And so most likely you're not going to go from infected seed to sclerotia to apothecia in the same year. And so, and furthermore, if you have your seed treated, and most, I'd say in Iowa, 90+% of the seed is treated, that's a really good way to have the infected seed not produce sclerotia. So the seed will still not germinate and produce a seedling, most likely, but oftentimes that seed treatment's effective enough to prevent the sclerotia from developing from that infected seed. So that's probably not a path that's likely. Most likely what happened is that inoculum was already there. And sclerotia can survive, a single sclerotia can survive, 5 to 8 years in the soil under some conditions. Sometimes it's just a year or two, but remember the mycelial growth that you have and the saprophytic growth that can occur from a sclerotia. These can perpetuate by producing more sclerotia. And so I've heard of fields where people have gone to 30 years of corn or to CRP or something away from soybean, come back to soybean, and they still get white mold. The sclerotia, the inoculum that's in a field, most likely will never go away. So really what happened probably was we just had the right conditions at this time for inoculum to be in the soil and to get that disease cycle started. Again, probably not coming from an infected seed, probably not coming from the spread of equipment, although, if you suspect those are the routes, you'd have to go back and look 3 years or 5 years in the past and see what was planted or what equipment was there to sort of lead up to this year. All right. Second question is like we had a drought in a lot of Iowa. We did not have a lot of rain in July, and so why did we get white mold? And so the possibilities would be, I'd say, like, it's just magic, which is obviously a joke, or we don't know what the heck we're talking about. But if you unpack the disease cycle a little bit, there's actually a pretty good explanation. And so if you look at when moisture is needed, it's going to be needed about, when you need moisture for seed to germinate to become a plant is when you would need moisture for the sclerotia to imbibe water and to start that process of producing an apothecia. And so early in the season, there was enough soil moisture for that to happen because we had enough for a soybean plant to grow. And so, yes, we didn't have rain in July, but the apothecia were most likely already starting to be produced. And these apothecia can survive. Once they're produced, they can be around and they can be producing spores for several weeks. The second thing is that, the cultivars might be more susceptible than normal. And so I can just speak up from, not pointing fingers at any seed companies, but I can just point at my program personally is the last 3 years, we haven't had any white mold showing up in our disease nursery that we're screening for the soybean breeders here at Iowa State and for a few others. It's just been really difficult. And so we haven't had a lot of valuable data to show what looks good or what doesn't look good. And so there's an increased possibility of some of these cultivars maybe being a little bit more susceptible than normal. So the other thing sort of is that we had such dry conditions that once we got those rains in early August, we actually had several of the soybean plants. They start to reflower. It's sort of a reset on on that crop. And when that occurs, what happens is that you've just extended the period of time when the soybean plants are susceptible. Remember, the apothecia are producing ascospores that land on that soybean flower. And so if you have flowering that's going all the way into August and early September, because the plant sort of restarted after the rain, uh, that then you have a really long period of time for infection to occur. And uh, so there's several different possibilities, and probably not all of them are going to be true in every field, but certainly the conditions did line up as far as early season moisture to produce the apothecia, and having a sort of a longer flowering period probably were the two driving factors for why we had so much white mold in such a dry year. All right, so now we're going to sort of talk on a little bit of the management. And so what is IPM, Integrated Pest Management, and certainly can it help with white mold? And I would say it's probably the only option, because there's not really one ticket for management of white mold. You're going to have to really hit this disease with several different hammers, and that's what integrated pest management is. And so the way I look at it is you're going to be picking strategies that are going to try to break up that disease cycle. And sort of the 5 ways that you can break up that disease cycle would be to find a practice that's going to reduce the sclerotia, the amount of inoculum in the soil. Some practices are going to prevent the apothecia from developing, or even maybe developing at the right time. So sort of they're going to be existing when soybean flowers don't exist. Sometimes you're going to have practices that open up the canopy, and that's going to reduce the environmental conditions, the humidity for the disease to develop. But this is also going to be reducing your yield potential because you have an open canopy. Cultivars are certainly one of the ways you can go. You can have resistance that slows infection and sort of minimizes the amount of damage that the fungus can have. But it is also, you can have sort of an avoidance mechanism where you have some kind of characteristic, like it's a fairly not bushy plant, a sort of an erect type characteristic, or maybe even planting a shorter maturity group, which would have a shorter flowering period, or maybe a plant that tends to not lodge. So lodging plants are probably the best thing for this disease. And so those are just either finding characteristics that are going to reduce the risk of getting disease, or maybe actually being resistant. And then fungicides certainly can protect the plant and protect that fungus from getting on the plant or spreading throughout that plant. All right, so let's go through just a couple of these, and really there's a long list. I think if you started going through all the literature, or even just talking to people, you can come up with a list of 10, 15 different things that could affect white mold. And we're not going to go through all of them, but what we can go through is just a couple of the newer ones, or some unique ideas, and maybe that these will shed a light on what other options are out there. But cover crops is something that has come up, and we know that cover crops can help with weed management. So can it help with white mold and sort of some other diseases? And there's actually some published papers out of Wisconsin that show if you rotate with winter wheat and have a wheat crop there, that is a way to reduce white mold. And then recently there's a group out of Cornell that published that they actually had cover crops that were folded over and remained out there, and those were reducing the amount of white mold in soybean. And so how does that happen? There's actually a couple of different mechanisms. The first one would be, we sort of have to go back and think about that disease cycle. You go from sclerotia, and if it's the carpogenic germination, producing the stipe, and then go into the apothecia. That sclerotia needs to be in the top 2 inches-ish of soil, because that stipe can't get too long. And so we're really worried about the top couple of inches, the sclerotia in the top part of the soil. Cover crops could just simply act as a barrier. And so instead of there being 2 inches of soil, now you have 2 inches of soil, and then now a little bit of some kind of crop. And certainly The wheat residue could do the same thing, or even a lot of corn residue has been shown to sometimes just act as an extra barrier, so that stipe can't actually get up to the soil surface. And that's important because that stipe needs to hit sunlight for it to produce an apothecia. Otherwise, it just stays a stipe. And so the cover crops could be simply acting as a barrier. The other thing would be they could be just providing enough shade. And so this is the really interesting thing about this pathogen is that stipe actually needs to not just be hitting light, it needs to be hitting shaded light, which would be predominantly blue light and sort of less of the red and infrared light. And so when an apothecia or the stipe hits the soil surface and there's shade there from either residue or this cover crop or whatever it is, they'll actually start to produce the apothecia. And in the case of that paper in Wisconsin on winter wheat, that's actually what happened was you had a lot of apothecia that were being produced really early in the season. So by the time you planted soybeans after it, a lot of the apothecia had already been spent or sort of used up all of their inoculum before the soybean flowers appeared. And so these cover crops can be sort of a barrier, but they could also be tricking that apothecia, tricking the sclerotia into producing apothecia before the soybean flowers appear. So this is certainly a possibility or something to look into. You do need a lot of residue out in that field for this to be effective. All right. Are there any biologicals that work? There certainly are a lot of claims out there, so hopefully one works. And really, there are some that are effective. And so Contans or Coney is a product that's Coniotherium minitans is the name of the fungus. So it's just a fungus that you're applying to the soil, and that fungus actually targets the sclerotia and digests them and uses that as their food. And so that is an effective way to break down or reduce the amount of inoculum in the soil. It's effective. I mean, there's been reports. Some of the reports are not up to 95%, so they sort of vary. But the best I've seen is 95%, and so you're reducing a lot of inoculum. But there are some limitations. It is a very expensive product, $40 to $50 per application, and a lot of times you need multiple applications. You also need to be putting this out at least 3 months in advance. A lot of times people put it on in the fall after a big white mold outbreak, or even really early in the spring, in hopes to have it out there with enough time to digest the sclerotia before the apothecia would be important. But these will not eliminate all the sclerotia. And again, think about that mycelial germination of the sclerotia and them being able to produce more sclerotia in that soil. And that could be an important mechanism to sort of make these biologicals a little bit less important as you sort of look over the many years that these products could be effective. And there are certainly so as far as the products that are targeting sclerotia, yes, they work, but they're expensive. And I would say really do your homework and make sure that they're going to be an economically viable option for your farm. As far as other seed treatments go, we evaluate them. We have them in our fungicide trials, and I'll show you some of that data in a few slides. But we really haven't seen anything else work as far as controlling white mold very specifically. All right, and this is the last section. So this is any recent fungicide data. As frustrating as it is, like we had a lot of white mold in Iowa this year. I had the only disease I had in my white mold trials this year was SDS. And so I struck out again, but I have colleagues in several different states that we do a standard test across all of the states, and they did have a good white mold development, and I'll show you that on the next slide. But remember that if you go to the Crop Protection Network and at the very end of this, you'll get a QR code for that. There is a fungicide efficacy pub, and in that publication, we rank all of the fungicides and the available ones that are out there as far as how effective they are. So I'll go through the treatments on this slide, because when we get into the graphs on the next ones, they're a lot smaller, but the text is pretty small and hard to read. But in general, we're targeting our three applications. The standard treatment that we're comparing everything to is Endura, applied at R1 and R3. And then we have either different fungicides, Omega, and then Delaro, Miravis Neo. And then some of these are actually applied with undercover sprayers, the undercover 360 sprayers, and or we are spraying them based on a predictive model called Sporecaster, and so we timed our application based on this app. And then we had Heads Up in there as well, and Heads Up followed by Domark, which would be sort of targeting very cheap options that are economically viable options that have been discussed as far as possible ways to manage white mold. All right, so our uniform trials were, again, 2022 and 2023, and we had 5 of them that had high white mold pressure. And so the data on the next slide will be from these 5 locations. So 2 in Michigan, 2 in Wisconsin, and then one in North Dakota. And again, the text is really small on the bottom here, but if you look at the second to last set of columns would be your non-treated control, which had a disease index of just over 40. And if you look at Heads Up as one of the treatments, the seed treatment, it did not reduce white mold at all. And in fact, that's the one that's to the right of the non-treated control. And then Heads Up with Domark is the one that's just to the left of that non-treated control. If you look all the way to the left-hand side, you can see the Endura applied at R1 and R3 is the one that's our check, and that's the one that had the least amount of disease. But as you go through it, you can see that there's 4 or 5 treatments in there that did a good job, and some of them were timed based on Sporecaster. So the 3rd and 4th treatment to the left were the ones that were timing using Sporecaster. So that's encouraging. And then also the undercover application was one that tended to help products like Omega, which are a little bit less systemic than some of the other products. And then so that the disease is the green, and then yellow is the yield. You can see that as you have more white mold, you get lower yields in general. And so it's just sort of a reversed relationship. So we're going to transition now away from white mold. There's certainly a lot of other things we can impact with white mold as far as opening the canopy and row spacing, plant population, tillage. There's all kinds of different things that could be affecting white mold. And certainly if you have questions about those, I would be more than happy to provide an answer. But those were sort of the most common questions I got throughout the year based on that disease. All right, so we're going to transition to sudden death syndrome, and this is a little bit of a repeat. So we've had a couple of big data sets that we're still diving into and looking at the data because they're that big, and so we're just given the last iteration of sort of where we're at with those. Just remember, this is a soil-borne pathogen, so we see a lot of foliar symptoms, but it is a soil-borne pathogen, and that pathogen stays in the soil. It never moves to the top of the plant. It's caused by Fusarium virguliforme here in Iowa. So there is a root rot phase, and then there's the foliar symptoms, and that how you get from root rot to foliar symptoms is that fungus produces a toxin. That toxin moves up to the top of the plant, and that's where you get the symptoms. All right, so the root rot phase, essentially it's just like Fusarium root rot. It is going to make the plant roots smaller and brown. And so there's nothing really too descriptive about that. It is a blue pigmented fungus. So at the end of the year, sometimes you can actually see that sporulation starting to come, and that would be an indication that you have SDS. If you can pull those roots out of the soil pretty easily, that's also another indication because the roots are just smaller. And then the foliar symptoms, hopefully this is a repeat for most people watching this, but it can start out as yellow mottling, pretty minor, but then it can very quickly develop into, this is where it gets its name, into this yellowing or intervenal chlorosis and necrosis, yellowing and death in between the veins, and then eventually you get defoliation. So management is just the IPM method, just like for white mold, is you're going to be hitting this with resistance. You're also going to manage soybean cyst nematode, because we know if you have soybean cyst nematode, that can be one of the ways to make SDS worse. There's several different cultural practices, planting dates, tillage, different things that you can consider that might affect SDS. And then making sure you have proper pH and fertility levels is also very important. And then there's some seed treatments that are fairly effective. So real quickly, just a quick update. We continue to do a product evaluation for SDS, and this has been now expanded. So it started out in about 4 or 5 states, and now we have, this last year we had it at 12 locations in 2023. And for this one, we're pathologists. We target fields that have a high risk of SDS. Sometimes we inoculate, sometimes we even irrigate. We do everything we can to make sure we get as much disease as possible. And then we have, again, a lot of site years. Just looking at the main list of products, and there's a couple of experimentals in here that I've taken out. But we have Ceramax, Elevo, Saltro. We have a combination of Murtec or TBZ, Heads Up, BioST, and a Scribe, and then a base seed treatment, and then an untreated control. If you just look at the data, I'm going to just focus on 2023 because I think I've shown 2021 and 2022 in the past, but in general, the base seed treatment and the base plus Ceramax and the untreated control were all around 18 to 20% FDX or 20 FDX, and you had about a 25% reduction if you used Elevo and Saltro, and then if you have that combination, the TBZ, Heads Up, etc., it did not really, that also was about over 20 FDX. And so there's only these 2 treatments, Elevo and Saltro, that are really affecting that yield. And then if you look at in 2022, I'm just going to point out in the yield in 2022, we did see several different products that had an increase in yield in 2022, although they weren't really affecting SDS symptoms, the foliar symptoms. But in 2023, it sort of went back to what we typically see, which is, if you look at the 2021 data, so the 2021 and 2023 match each other, where Ceramax, the TBZ, Heads Up, etc., did not really affect the yield compared to the base or the non-treated control. But we did see that 2 to 3 bushel increase using Elevo and Saltro, and there are no differences between those products. And so we continue to look at products that are targeting SDS, and again, my encouragement, anyone watching this, if you've seen advertisements or you have things that you want us to try, please let us know, and we'll get it added to our evaluation for 2024. So the study that's been updated is that we had this larger risk study where we had 123 locations across much of the U.S. and Canada, and we were targeting fields that had both the fields had susceptible and resistant varieties. Most of them did, and then they had either a base seed treatment and then a base plus Elevo and a base plus Saltro, and then we were collecting foliar symptoms, root rot, yield, and then a lot of soil sampling. And that soil samples went to Michigan State for FV levels, and then we did SDN counts, and we also did soil analytics, soil DNA analysis with pattern ag, and then we also collected weather conditions. And so not only were we collecting soil samples, but we were also getting a lot of the soil characteristics off the map. And so Real quickly, just to show you, most of these are in the Midwest. A lot of them are in Iowa, Illinois, Michigan, Ontario, and then sort of the neighboring states. Again, we had 123 locations to start with, but not all of them had SDS. Not all of them collected all of the data that were required. And so as we were going through the analysis, you actually see that number start to drop a little bit in subsequent slides. So don't be surprised. But some of them had a lot of SDS, and then some of them had a lot of soybean cyst nematode as well. And so, again, we were sort of targeting fields on purpose that would sort of be this whole range of high risk, low risk of SDS, high risk, low risk of soybean cyst nematode. So overall, we had very little effect across all locations we used in a resistant or susceptible variety. We had a little bit less yield with the resistant variety, but it's really splitting hairs. And then if you look at the base Levo and Saltro, there's a small percentage, 1.5 to 2% increase using Levo and Saltro. But again, when averaged across all of the fields, some of them did not have a lot of SDS. That difference wasn't really that significant. But if you start breaking them down as looking at, okay, you had no FDX, no foliar symptoms, or less than 10 or greater than 10, you can see that you start to see differences. And so if you look at the graph on the left-hand side, the resistant variety actually had less yield in the fields that had no FDX, but they had more yield in the fields that had greater than 10 FDX. And if you look at the seed treatments, the differences in Saltro and Levo compared to the base, they were significant, or actually increased a little bit in the FDX is with no disease or the less than 10. But you can see that the differences were more pronounced when you had greater than 10 FDX. And this is very consistent with what we see in a lot of our published research, is that, and it makes sense. The more disease, the more effective that seed treatment is going to be, or the more needed that seed treatment is going to be. And so in those fields that had greater than 10 FDX, that's when you see the resistant variety and the seed treatments being the most useful. So if you look at the FDX category of just less than 10 and greater than 10, if you look at the amount of FDX, and now we're not looking at yield, we're looking at FDX, you can see that both the resistant variety did a great job of being effective and having less disease, the seed treatments as well. And so you had these products work, and again, we've shown that, and in the previous slides, you had the yield responses that would go with it. And this is what's really driving that yield responses, the ability to reduce the amount of foliar symptoms being produced, both with the resistant variety and the seed treatments. So the ultimate goal, what we are trying to get to, is like, can we identify fields where we can have an increased chance of saying, yeah, seed treatment is going to be needed in that field. And so essentially looking at the risk of FDX developing in a particular field. And we really got we're pretty excited because there's a paper that was published in 2019 that showed that you can actually go in and start looking at and predicting the amount of symptoms that are going to show up based on the amount of inoculum that's in the soil or the amount of SCN that's in the soil. Again, remember, soybean cyst nematode is going to increase the amount of symptoms. So this paper is it's just one location in one state, but it's given us an idea that, hey, we can actually go through and come up with some kind of workflow that would identify the risk. And so we have soil sampling. You can also just collect field data and go in, extract DNA, go through the qPCR, and it all goes into this data analysis, and that will give you the risk. So I had a graduate student that was just looking at the field data side of things. Michigan State was really focused on the soil sampling side of the top tier of that graphic. So from these 123 fields, and then eventually a little bit less as we started looking at what data was collected, we broke it down into 4 different packages. And so one of them being farm management, which is the category 1. That's going to be your resistance to the in the variety. Did you use a seed treatment, planting population, planting date, seeding rate, row spacing, irrigation, tillage, and previous crop? And so things that going in ahead of time, and is sort of on we call it the farm management level. The second one is soil factors. And so these, again, are things that you can collect and know ahead of time, just with the soil sample, or just diving in and looking at the soil type, online. But having your SCN populations, you can have spring or fall, soil type, organic matter. Again, sort of you can read the list, but these are all things that you can collect. If you pay a little bit extra money, you can actually get an idea of what the microbiome level is. There's several companies out there that do this, but we use Pattern Ag, and so we took a lot of the soil samples and we sent them to Pattern Ag, and we got data on all of these different things here. So again, but they're also looking at SCN or Fusarium. Remember, this is caused by Fusarium virguliforme, and then a lot of other factors as well. And then the last one is just a lot of weather variables. And so this is going to be mostly centered around temperature and soil moisture or rainfall during very specific times of the year, to see if we can get an idea as to when these rain events or the weather conditions are going to be affecting SDS development. So what we did is to pick out which variables we actually wanted to use. We used only 84 locations that had the most data collected, and then essentially we did a correlation, and then we threw out anything that was not in that red box. And so which meant that if it was outside of that box, it was actually too related to something else. So for example, if I'm just going to make something up on the top of my head, but if you had soil moisture at 1 to 2 weeks, and then you had soil moisture from 1 to 4 weeks, those might be so correlated that it didn't make sense to include both of them. So we'd only include one of those variables. And so we eliminated a lot of the variables that fell outside of that 0.8 or -8 to 0.8 correlation. So we took all of the variables then, and then we actually use 4 different models. So we have a logistic regression model, random forest model, recursive partitioning model, and XG boost model. And we took all of those variables, and then from the 4 packages, and we just kept adding packages. And I'm going to just show you the data. If you look at the accuracy for packages 5 and 6 on that table on the bottom left, so there's a lot going on here. But essentially what it's showing is that if you took package 5, which is the cultural practices, the sort of the farm management packages or practices, and the soil health, not the microbiome, but the ones that just take a soil sample and then know your soil data, you have an accuracy up over 90% of predicting if these conditions would lead to having more SDS. And then package 6 was adding weather. You can see that the accuracy didn't really change that much. And so, having there, and then what we did was actually look at all 4 models, and then we ranked all of the variables that were within each model, and then selected the ones that were the most relevant. But if you look here, the 5 and 6 sort of continually were near the top as far as accuracy from these different models. And so we're just focusing on packages 5 and 6 for the next couple slides. And so if you look at just what came out of that, or the risk factors, and this is sort of for me, the most valuable slide was the pH. If the pH was greater than 6.5, then SDS increases. Organic matter, or if it was greater than 1.5, we had an increased chance of SDS. Precipitation early, so this would be sort of leading to that root rot phase. And then soybean cyst nematode, which we've known, so it's good to see that that's sort of shown up as a risk factor, because we knew that was from literature in the past, that's something that's there. And then the precipitation during the reproductive stages. which makes sense because that's where you get the toxin sort of being flushed from the roots up to the top part of the plant. Seed treatment, and then also sand was ended up on the top 7 list. And so, and these were all again weighted across all 4 regression models when we're looking at this. So then, the next step. So that I'll just go back. So this is just the for me, an interesting, this could have been the stopping point. We could just say, okay, here's the things that we found to be the most related to there being a chance for these to increase the amount of SDS. And some of them you can't affect. You're not going to be affecting the amount of precipitation at certain times of the year, and we certainly can't manipulate the soil types. But these will give you an idea, like, hey, if I'm planting into these soil types and I have soybean cyst nematode and my pH is higher than 6.5, these are the conditions that might lead to needing a seed treatment. So one of the things that we did is we actually went back to the recursive partitioning and we removed seed treatment, because what we wanted to try to do then was figure out, is there a situation where A, B, and C line up where a seed treatment could work? And so we actually did a new analysis. We removed seed treatment from the analysis, and then we're going after, is there a time when a seed treatment could be needed? And so so the list changes a little bit when you remove the seed treatment from the analysis. And then essentially what you do is you go through this dichotomous key. And so I'll just real quickly, if you have what happens when you do recursive partitioning is that it identifies that point, sort of the tipping point, where if you get past that, then the risk is higher. And so so for in this case, if you had pH greater than 7, then or less than 7, and then the key starts. And then if it's less than 7, then you have a low risk, and then you go over to precipitation, and it sort of breaks it all out, where you have the sort of low and high risk. So essentially what we're doing is we're taking all of this recursive partitioning and we're going through and using the top 8 risk factors from these as we're going through and now reanalyzing the data. And I'm just going to use the example of the soybean cyst nematode greater than 2,000. So that's one of the recursive partitioning points. And does that affect SCN? And so if you look at the graph on the left, that's the yield. You can see that less than 2,000 Salter and Levo, again, had a little bit higher or even with the base seed treatment. And then if you get into that higher than 2,000, actually Levo looked a little bit better than Saltro as far as yield goes. If you look at the amount of FDX, you can see the less than 2,000 and then greater than 2,000. You can see that there are some differences. And then if you just really focus in on SDS greater than 2,000 with disease, you can see with greater than 10 FDX, you can see that the seed treatments really did work, reduce the amount of FDX. And there's maybe some, at least not statistically, but there were some numeric differences between the seed treatments. And so we're in the process of redoing this recursive partitioning for all of the factors, and so sort of stay tuned on that, and hopefully in the next couple of weeks we'll have that done. But just wanted to give you an update on where we're at with these. So summarize SDS. Again, remember there's two phases. They both contribute to yield loss. Resistant cultivars are the best. We didn't talk about resistance, but that continues to be the number one way to manage this disease. But you can add a Levo or Saltro to both a resistant variety or even a susceptible one, and they're going to improve or protect yield across all of it. If you have higher FDX, seed treatments are going to protect more yield, which makes sense. And they even, at least in this last year, they did a little bit better job than the resistant variety that we selected. And so really the last bullet point is we are continuing to try to identify fields that have greater risk, but I'd say really focus on right now. It looks like pH, organic matter, and soybean cyst nematode are the ones that you really want to go after, and hopefully we can provide more details as to what exactly that looks like and how that would affect the use of a seed treatment or not. With that, tons of people have supported this, and you always run the risk of throwing out a lot of logos. As I'm looking at it, I know that North Dakota State provided a lot of the white mold data as well. But just kudos to a lot of my colleagues, Wade Webster, Damon Smith, Marty Chilvers, etc. And a lot of the soybean checkoff boards are extremely useful, making sure that we have the funds in place to be able to do these studies. So with that, I promised that I'd show you the QR code for the Crop Protection Network if you wanted to find the results to the foliar fungicide efficacy tables for white mold. We also have one for seed treatments for SDS. So more information from the Crop Protection Network, you can go there or just go to cropprotectionnetwork.org. And with that, thank you so much for listening.